Hi, everyone. I'm super excited to be here today and introduce Interface Biosciences. We are a platform drug discovery engine defining the future of microbiome therapeutics. My name is Hannah Wastik, and I'm joined by my co-founder, Will Van Churen. Together, we started Interface Bio in 2020 based off a collective 10 plus years of research done at Stanford. Both of us completed our PhDs studying the microbiome, which is the collection of trillions of microbes living on our skin and in our guts. An absolute explosion of research in the past 5 to 10 years has implicated the microbiome in nearly every organ system as well as drug metabolism. Because the microbiome plays such a huge role in nearly every inflammatory disease in the 21st century, it lends itself as an enormous opportunity to create drugs or therapeutics by better understanding the microbiome. Unsurprisingly, this sector has attracted significant venture capital dollars, cresting over 5.4 billion in the past decade alone. Even without approved drugs, microbiome companies are securing billion-dollar deals with pharma partners. And they're all using the same approach. They're all giving live microbes as drugs. These are also known as live biotherapeutics, and they have some serious risks attached to them. The first is a limited discovery process due to noisy samples and a small search space. Second are some serious commercial risks, including high expense to manufacture, unpredictable efficacy, and a reduced patient pool relative to small molecule therapeutics. The reason all of these companies are so limited is that they're looking they're missing the most important part of the picture. When I showed you this picture before, I used a level of detail that people focus on today. Microbes create some sort of human cell response. What I didn't mention is the most important part and the reason why these companies are limited to correlations. The most important part are the chemicals or the metabolites that microbes produce. These chemicals are small enough to soak into the skin, pass through the gut, where they enter our systemic circulation and exert their effects on human cells. These metabolites are the language by which microbes use to communicate with our human body. These metabolites are the key from going from correlation to causation, the key to truly understanding how the microbiome interacts with our body. And we have the solution. Interface Bio is the only company to be able to focus on these metabolites instead of microbes. Our unique platform is able to decode this language to find the chemical reason that microbes are able to help people and transform them into therapies for many different diseases. Let me show you how we're doing this. We have a deep tech pipeline that is experimental and computational. We create novel data sets and bridge them with machine learning. This is simplified into 3 steps. First, grow to build our data moat. Second, featurize to annotate the microbial products that are being produced. And finally, assay to measure the effect that these chemicals have on our immune system. Put another way, we create these complex biological data sets, transform them into data frames that are parsable using machine learning, then feature select which of these microbial products are causal for immune modulation. So let me show you how we're doing this in the lab today. The first step is to grow. This is our most important step and the reason why no one else can do what Interface Bio can. Currently, microbes are grown in standard complex medias, and this is problematic for 2 reasons. First, they kill human cells. Second, these standard medias inhibit measurement of the microbial products. This means you can't create the data moats I was talking about, and you also can't assess the therapeutic efficacy of these chemicals either. Interface Bio's proprietary media is better than standard media for 2 reasons. One, it grows microbes better. That means they grow more and they grow faster. Next, they don't kill human immune cells. With these 2 key bottlenecks alleviated, Interface Bio is the only company to be able to grow these microbes at the industrial scale and assess their chemicals for their therapeutic efficacy. Our second step is to featurize. We have a state-of-the-art metabolomics pipeline developed at Stanford, enabling high resolution of microbial chemistry. We have 500% increased sensitivity compared to standard methods, meaning we can measure more of these chemicals. Our last step is to assay to figure out which of these chemicals are causal for immune modulation. And I'm so proud of this data because it's the first experiment that we did. We've already found a hit, a microbial chemical causal for decreasing inflammation. And this is just the start. We believe there are so many microbial chemicals out there just waiting to be developed into drugs. And the first place we're going to do that is the most common inflammatory skin disease in the world. Atopic dermatitis affects 9.5 million children and 16.5 million adults in the United States alone. First pass therapeutics are topical steroids, which can only be used for about 2 weeks. Second are injectable biologics such as Dupixent, which are only effective in 37% of patients. Excitingly, Interface Bio already has preliminary data showing a suite of potential therapeutics that promote wound closure for the treatment of atopic dermatitis. We're going to build on this momentum in the next 24 months and focus on 2 inflammatory diseases, atopic dermatitis and psoriasis, to build our platform quickly and organically. Our key deliverables at the end of this are a lead asset ready for IND in clinical trial and another asset in the pipeline already advanced through preclinical disease models. We're going to achieve this ambitious vision with a world-class team hailing from the best research labs and institutions in the world. We're also guided by a scientific advisory board with micropost and clinical development expertise. We have the best technology, we have the best team. The time is now to define the future of microbiome therapeutics. Thank you.